Hello. Thank you very much for listening to my presentation, Citizen Science is Filling the Forage Data Gap for the Chesapeake Bay. My name is Caroline Troy, and I am a junior at Brown University studying environmental science. Last summer, I participated in an internship with Morgan State University's Patterson Environmental and Aquatic Research Laboratory, or PEARL, located on the Patuxent River in Maryland. My mentor was Dr. Thomas Eide, and for this project, I assisted him and worked to further his project focused on collecting forage species data through citizen science. So, let's start with some background on the subject. What is forage? In a marine ecosystem, forage is considered to be small fish or invertebrates that are prey to many predator species. For example, forage could include blue crabs, grass shrimp, perch, or mummichogs. Forage could also include young fish from larger species that are important to fisheries when grown, for instance, juvenile weakfish and Atlantic croakers. All these forage species are an essential part of the bay ecosystem, as they support so many larger species. However, despite the fact that so many species rely on them, forage species and what habitats they are most closely associated with are rarely researched, as their study is generally viewed as a lower priority to fund than that of larger marine animals like striped bass that have a direct influence on the economy. However, it does not make environmental or economic sense to neglect to study these smaller species and younger fish, as their dynamics and trends have a powerful influence over the bay's health as a whole. During my internship in summer of 2019, I had these three goals to help move the project forward. First, to collect data about forage species in varied habitats in Jefferson-Patterson Park on the Patuxent River where PEARL is located. Second, to analyze data to look for variations in species compositions across different habitats and conditions. And third, to involve local monitoring groups in the project to expand its reach while educating citizens who have an interest in their local waterways about forage. Some examples of the kinds of habitats we are interested in sampling include areas with submerged aquatic vegetation, areas adjacent to Spartina or Phragmites marsh, habitat with woody debris, areas bordered by bulkhead or riprap, and mud or sand bottom habitats. So, to start off, I'm going to describe the project's methods through my work collecting samples at PEARL during summer of 2019. I began with sampling 9 sites within the park's boundaries. These 9 sites included 6 different habitat types. I set and collected unbaited minnow traps bi-weekly, with 1 to 4 days between trap set and collection. Minnow traps were originally selected to be used in 2017. They do not require bait, and they are easy to place and check, compared to seine nets or other sampling techniques the citizen scientists may need more extensive training in if they were to complete the sampling correctly. As you can see on this map, here is PEARL's headquarters, and here are the 9 sites I sampled. We had a variety of habitat types, from more developed habitats like riprap to marsh sites, as well as woody debris. And here you can see photos of what some of those sites looked like at the park. Now I'm going to talk about the process of setting these minnow traps. Setting a trap requires a written data sheet, which I redesigned to incorporate clear instructions in order of data entry and with multiple choice options when possible. The data sheet requires information on the time, habitat type, weather conditions, trap depth, and water quality information, such as temp, salinity, dissolved oxygen, and water clarity at the trap site. These parameters can be measured using a Secchi disc and a water quality sonde. In this photo, you can see me taking water quality data and a fellow intern, Sabrina Tolbert, noting the results. After recording all the data, we, and our citizen scientists, place the traps. Next, I'll explain the trap checking process. A full day's set ensures that the trap sampled a full tidal cycle. Water quality and weather data conditions are recorded again, and the traps are pulled. Forage species and numbers of each are recorded. Images are taken for species whose identification was uncertain for later confirmation. In these photos, you can see better what the minnow traps look like. There's a simple latching door mechanism and a funnel entrance for the catch. You can see in this photo, we've managed to catch an American eel. As I mentioned previously, the final step of the data collection process in the field is recording the catch before releasing them. Here are some of the photos of the species we caught. We often found mummichogs, American eels, and blennies. We also sometimes found young fish directly valuable to the fishing industry, such as this spotted sea trout seen here, which are an important recreational species. Throughout the internship, while working to collect new forage fish data for the park, I compiled all the written data sheets collected by the 4 citizen science groups in 2017 into a common spreadsheet. The 4 groups engaged in the project in 2017 were the Havre de Grace Maritime Museum, the Midshore River Keepers, Severn River Association, and James River Association, whose supplies for the project were funded by the Chesapeake Bay Program Office and the NOAA Chesapeake Bay Office. These 4 groups sampled in distant regions, giving us a wide window into species variation along the bay. At that time, combined with my own contributions from the summer, there are around 300 samplings total to compile into the forage fish spreadsheet. Thanks to Caitlin Ritchie of Pearl, who used latitude and longitude locations of each sampling site to create an interactive GIS map, one is able to look at the exact locations of each individual forage fish sampling site from previous years. And as you can see, if you click on a gray data point, you get a window that tells you about the species caught, the location, the water quality, who collected it, and all the information that can be found on the original datasheet. This tool helped us better visualize which area of the bay need greater sampling efforts and allows for an understanding of regional trends. After compiling the datasheets from all the sampling in the bay completed at that point, I was able to do some basic analysis to look for trends in our number of fish or numbers of invertebrates that could be explained by factors such as duration of trap set, water quality parameters like water temperature, salinity, dissolved oxygen, percent water clarity, habitat type, or the time of day. Of these analyses, our graphs on trap set duration were some of the most helpful. As you can see in these 2 graphs here, the first shows the number of fish caught against the duration of that particular trap set length. The second shows the same for invertebrates. From these graphs, we found that vertebrates reached trap capacity at about 3,000 minutes, or roughly 2 days. Invertebrates reached trap capacity at about 1,500 minutes, or roughly 1 day. So we concluded that 1-day sets are probably sufficient to get reasonably representative samples of both vertebrates and invertebrates. This information helps us refine our sampling procedure and increases the frequency at which data can be collected. For the parameters other than duration, there were several other interesting graphs, but most of the relationships found in these graphs weren't strong enough to convince us that these relationships weren't just due to chance. We simply need more data, especially since a lot of our data ranges are limited in season and place. Another thing we could do with more samples is break these relationships down by the species level, which would also be expected to show interesting trends. After looking at the trends for all of the bay data we had collected, I then decided to focus in on the data I collected just in Jefferson Patterson Park that summer. So, using just the trap data I personally collected that summer, I also analyzed the data to look specifically at the average number of fish, or the invertebrates, caught in a trap in each habitat type. This orange graph is of the average total fish caught in each habitat. You can see we had our highest average numbers of fish in traps in woody debris, Phragmites, and Spartina grass marsh habitats. The majority of fish logged at the park were mummichogs, or Fungulus heteroclitus, and this was somewhat surprising, as numerous studies have found the abundance of juvenile F. heteroclitus to be significantly lower in Phragmites marsh than in native Spartina grass marsh, but we found the opposite. One factor could have been that much of our marsh areas, though predominantly either Phragmites or Spartina, were mixed. The yellow graph illustrates the average number of invertebrates caught in traps in each habitat type, and clearly the highest average number for invertebrates was in the Phragmites marsh, and to a lesser extent riprap and woody debris habitats. The next step I took with the park-specific catch data was to analyze the data by species and create a graph that illustrated the percent composition of species in each habitat type. The riprap habitat had the greatest diversity of species, and to a lesser extent the Phragmites and woody debris habitats, while the sand bottom habitat had the lowest diversity. This low diversity in the sand bottom habitat is notable, as many state juvenile fish surveys use a seine in sand bottom areas. As our results suggest a low forage diversity in this habitat type, these surveys might not be a good representation of forage abundance, although they may better reflect relative abundance of harvested juvenile fishes. However, our work so far is a very preliminary analysis, and larger sample sizes will help lend more confidence to these comparisons. Another preliminary observation I found was that the highest composition of mummy chogs was primarily in grass, Phragmites, and woody debris. The finding that woody debris could be valuable forage habitat is intriguing, as very few papers sample in woody debris, and our results suggest the usefulness of our minnow trap sampling method for this potentially valuable habitat. As I mentioned before, another large part of my project was reaching out to and meeting with several Chesapeake Bay groups. Last summer, we made positive steps to continue citizen science sampling on this project with 3 groups, and met in person with 2 of the groups. The Magothy River Association, one of the groups that received an in-person presentation to demonstrate sampling techniques and an outline of commonly found species in the traps for their region, has been particularly active. They began sampling last fall and have continued through this spring and summer. In the case of this group, once they had the initial supplies and instructions, their volunteers, who are often waterfront property owners who could easily set traps, were comfortable to continue sampling throughout the rest of the year. This project is clearly in its beginning stages, but with the enlightening results already seen from our preliminary analysis, more sampling with citizen science groups around the Chesapeake Bay should be completed. Here are some goals for the future of the project. First, as I said before, we hope to engage more citizen science groups. This increases the number of observations, expands the project's spatial and temporal representation, and involves and educates more citizens. Additional data will provide improved analysis on the effects of parameters, such as set duration, season, water quality. And eventually, we hope to examine the data to find those species and habitat relationships. More data will allow improved resolution of species assemblages in each physical habitat throughout the bay system. Finally, results from this project will inform fisheries managers, inform policy, and protect essential habitat for forage or young fishery species in the Chesapeake Bay region. To finish, I would like to thank my mentor and co-author Dr. Thomas Eide, as well as Richard Lacouture, Caitlin Ritchie, and Director of the Pearl, Dr. Scott Kanaki, the project's community partners, along with the other interns who assisted me, and everyone at Pearl for this opportunity and their continued support. I'd also like to thank AFS for the opportunity to present this project. Thank you.